Does this work? Yeah.
Yeah, looks good.
Okay, great.
How's everything going?
Good. Busy, but good. How are you doing?
Doing well.
Where are you now?
I'm in Shanghai.
Oh. So now you can travel more easily now, I guess.
Yeah.
That's nice. So how's the New Year's in?
I just came yesterday to Shanghai, but I was
I see.
Yeah, I was back in California. Actually, I went to South America a little bit for New Year's.
Oh wow, where in South America?
Actually Panama, so Central America.
Oh, that's nice.
I planned a whole, because I was just back by myself, so I planned a trip to Hawaii with my college roommate by myself, and then my whole family got COVID in China, and then so they bailed out and they came back early, and then they didn't want to go to Hawaii, so we went to Panama instead.
Oh, okay, that's a nice substitute. So this was when the COVID was spreading in China, or?
Yeah, it pretty much spread like within 1 or 2 weeks.
Karen.
Hey, it's all.
Carina.
Hi, nice to meet you.
Nice to meet you. Miriam.
Hey.
Good to see you.
We should try to get together, it's long.
Yeah, definitely. So
I just got back yesterday.
Yeah. So I got all the in-person meeting in China. We go down as well. We have some cell biology meeting in March or April, I guess. It's postponed from last year.
Okay.
Somewhere Suzhou. Yeah.
Okay.
Yeah.
I'm organizing the ALS meeting. We tried last year again. I have to settle on a date, but I'll let you know.
Sure, fine, definitely.
I paid like 30,000 renminbi for the dinner. I hope they still remember that, okay. And the restaurant's not out of business.
This deposit should be fine, definitely. Yeah, it will give you a good upgrade to the fancy dinner.
Yeah. No, this wasn't a deposit. I paid for everything in advance.
Oh, okay.
That could have been a mistake.
Hopefully the restaurant will still be there.
I know. I hope so.
Yeah. Since I come back, Shanghai is all the restaurants open. People has the movie theater is open. Yeah, everything come back.
Hi, Jen.
Hi, Jen.
Hi.
Hey.
Hi, Miriam.
Hey, good to see you.
Good to see you. Well, Aaron, you're just flying over.
Yeah. I feel like we were just meeting in San Diego.
Yeah.
Oh.
Is it raining in Shanghai?
Oh, it's beautiful.
Yeah.
It's my apartment. Yeah. It's another beautiful day. Cold, but good.
We just got another rainy day.
Oh, raining.
Yeah, so much rain this year.
Great.
Yes.
Have you told news, Aaron?
I'll say something. Yeah, I'll say something.
Sure. Yeah.
So Shanghai, everything is all back, work, holidays over.
Yeah. So is people back to work, and all the restaurant, movie theaters come back, everything come back, luckily. Yeah.
And
my family wants to go to a movie, but I don't like going to movies normally. But
Yeah, there were a lot of nice movie going on in the New Year Day.
Okay.
Should-
Yeah.
Should watch one or two. They are just like summer season in US. There are a huge amount of new movies, especially try to have some marketplace.
I'll take him to watch Chinese movies.
Oh yeah.
Okay. How was your meeting, Zolong?
I postponed to next week to be safe. I don't like the tight schedule, so I said okay.
Okay.
That's it. Next week, and next week I will put the meeting after Neurozoom.
Okay. Good.
8 early meetings. Yeah, make me nervous.
They make me sleepy.
I like early meetings.
You must be a morning person.
Yeah,
I'm both.
Yeah,
Both morning and night person.
getting up early will kill me.
I'm with you, Zolong.
Yeah.
Since I couldn't stay late either, so.
Should we get started, Zolong?
Go ahead.
All right. Hi, everyone. Welcome back. Happy Chinese New Year. Good to see everyone. Shanghai edition of NeuroZoom. Eagles going to the Super Bowl edition, NeuroZoom. Good to see everyone. Looking forward to the talks coming up. And before we get started, one second. Before we get started, I want to advertise the next set of talks coming up next week. We have Cui Wang from Stanford and Mark Rossi from Rutgers University. And brief NeuroZoom announcement. As the saying goes, all good things must come to an end. So NeuroZoom will be ending soon. 3 more weeks. So it's the final countdown. It's been a lot of fun. So we have Cui and Mark next week. Then the penultimate talks will be from Dan-Chan Liu at ION and my best friend at Stanford, Jin-Young Lee, electrical engineer, neuroscientist. She'll give a talk. And then for the grand finale, me and Zolong will give a little science update from our labs and talk about other things. We'll have more to say about NeuroZoom, but it's been really a lot of fun to, there's like 268 talks we've heard over the last 3 years. And it's been great to be able to meet all of you, hear great science. It's always extra special when I see some paper come out from one of you that you've talked about a few months ago here. So that's been exciting. It's helped us get through some dark times, helped us stay connected. We'll have more to say on it, but thanks again. It really has been a privilege to see everyone. Okay, with that brief announcement, please get ready for our great talks from Carina, Hanashima, and Miriam Goodman, and Zolong will introduce Carina.
Okay, excuse me. Okay, it's my great pleasure to introduce Professor Carina Hanashima from Waseda University. So Carina has get her training in early from Waseda and later on Memorial Sloan Kettering, and as well as third board in NYU with Goodman Fischel. So after she come back to Japan to set up her own lab early in Riken and now in Waseda University. Carina has a study focused on the molecular mechanism underlying the cortical development. So today we'll talk about the cellular and molecular mechanism underlying assembling of neurocortical cytoarchitecture. So welcome, Carina.
Thanks,
Great.
Dilong, and thanks, Aaron. Let me share the slide. Is this working?
Sure, great.
Okay. Okay, thanks again, Aaron and Zilong, for organizing this seminar series. And I was just about to say that hoping that this series is going to be continued, but now I understand this is going to be the countdown. And I pretty much appreciate both of you for this wonderful, scientific interactions. And now we can actually meet in person to discuss more about future science. So for today's talk, I'm going to talk about the neocortical development, as Zilong said. And our laboratory has been focusing on using these mouse models. And I guess many of you may be aware of that the neocortex is a specific organization to mammals. And if you look into the detailed cytoarchitecture, meaning the different cell type organization, using this mouse brain, this is a coronal section of the mouse brain, you could appreciate that these neocortex is organized into different layer structures in which each of the layer has unique cell expression repertoire, as well as connectivity. So here I'm showing you the layer 1 Cajal-Retzius cells, which expresses reading. And then these intracortical projection neurons expressing brain 2 in the layers 2, 3, and also the thalamic input, the sensory input neurons, which expresses aurora beta, and then finally the deep layer projection neurons, which express CTIP2. So these are the neurons that project far away from the cortex. So it's been appreciated that these neocortex, both in mouse and in humans, are organized in 6-layer structures. However, if you focus onto these different 3-layer subtypes and take a lower magnification view, you would also see that these layer structures are not just organized evenly among the different areas. However, there are differences in the layer thickness. For example, here I'm showing the aurora beta, which is abundant here, and then which is almost missing in this medial portion of the neocortex. So you could view the 3D cytoarchitecture of the neocortex in which the layers are organized from different subtypes. However, these layers are organized in distinct functional districts. And it's been appreciated already back in 1909 by Goodman that the cytoarchitecture could represent different functional information processing. For example, the area that are abundant in layer 4s are primary sensory areas, and areas which lack layer 4s are the motor areas. So the laboratory has been interested in understanding how the different subtypes of these neocortical, these are especially the glutamatergic excitatory neurons, are specified and then organized into these 3-dimensional structures. And for the past years, we have been mainly focusing on understanding how these laminar subtypes are generated from the common progenitors in the neocortex. So back early on in development, these neocortical neurons are generated from the common progenitors residing in the ventricular zone here in the dorsal part of the telencephalon. And these cells undergo substantial proliferation, and then at one point they started to divide asymmetrically to produce one neuron and then one stem cells, progenitor cells. So if you follow the neurogenesis of these cortical neurons, initially in mouse at embryonic day 11.5, the progenitors start to produce the earliest neurons which expresses reading. These are the Cajal-Retzius cells in the mouse neocortex. And then 2 days later in development, you now see these CTIP2 positive deep layer projection neurons being generated below these Cajal-Retzius cells. And then finally, 2 days later, you see these brain 2 expressing progenitors, which undergo mitosis and become postmitotic, and then migrate past these deep layer projection neurons and then occupy a more superficial position. So you could view that these neurogenesis of these cortical neurons progress in a sequential order in which the progenitors undergo distinct phases of temporal complex and undergo asymmetric cell division, in which one progenitor cells first produce Cajal cells and then sequentially produce deep layer neurons, and then finally the upper layer neurons. So we've been interested in understanding how the temporal transitions between these major subtypes occur during the cortical development. So initially, we have focused on how the transition between the earliest Cajal cells to deep layer projection neurons occur. And for this, we have focused on one of the 4K transcription factors, FOXG1, which is known to be a robustly expressed transcription factor in the developing forebrain. It's actually a marker for the telencephalon. So you could see early on the LAC-Z knocking heterozygote of FOXG1, which is representing the whole telencephalic property in the developing neural tube. And when we look into the development of these the cortical neurogenesis in these FOXG1 knockouts, initially the onset of Cajal neurogenesis, meaning that the reading expressing Cajal cells progress normally. So you see these reading positive cells around E11.5, similar to the heterozygote controls. However, 2 days later, and sequentially, if you look into the neurogenesis of these FOXG1 knockouts, instead of undergoing these sequential deep layer to upper layer neuron productions, in the FOXG1 nulls, they keep on producing these earliest born Cajal cells. So if you see it in the scheme, instead of the transition of these Cajal to deep layers to upper layer neurons, in the absence of FOXG1, these progenitors are no longer able to undergo these temporal transitions. So we did a series of studies, and I think the most simple one to show here is when you do electroporation of FOXG1 in the FOXG1 constitutive knockouts at the time point in which upper layer neurons are normally born in controls, you could see that these GFP positive cells, which are now expressing FOXG1 in these Cajal cluster in the FOXG1 knockouts, and these FOXG1 positive cells are now able to switch from making these Cajal cells to the deep layer projection neurons. And Takuma Kumamoto back then did a series of studies using transcriptomics and ChIP-seq and conditional mutants, and identified that FOXG1 alone is sufficient to repress up to 20 transcription factors, and then switch the program from making these early Cajal cells to next sequential deep layer projection neurons. So now we next were interested in how the transitions between these deep layer projection neurons to upper layer projection neurons are instructed. And for this, we demonstrated that this transition requires a feedback from the post-mitotic neurons from the deep layer neurons themselves onto the progenitors. And for this, Kenichi Toma back then did a series of studies, and one of them was doing the genetic ablation of deep layer neurons in vivo. And what he did was to cross these neurogeny to Kriar mice, which enables to recombine in the early post-mitotic cells, and then combine that with the Rosa-Stop DTA lines to kill off the early born neurons. And by giving tamoxifen at E11, 12, 13, you're now covering the entire deep layer neurogenesis phase. And if you analyze the cortex at E14.5, you now see that there is a substantial reduction in the number of the post-mitotic deep layer neurons being produced, as indicated by the combinatorial marker of CTIP2 and ZFPM2 covering the layers 5 and 6. So now you're killing off a substantial number of deep layer neurons. And when we analyze this cortex, deep layer ablated cortex at E18.5, right before birth, what you would expect, because you're now killing off the deep layer neurons, you would expect that their upper layer neurons are being overrepresented. However, when he did the quantification, he sees that the ratio between these deep layer CTIP2 expressing portion and the SAPI2 expression upper layer proportion was not significantly different, meaning that there is some kind of a compensation in this deep layer ablated cortex. And he further went on ahead to do a birth date analysis by giving a pulse of EDU injection after this ablation of these deep layer neurons. And now he sees that instead of producing the upper layer neurons, which are supposed to be the normal birth date at this E14.5, now he's seeing a substantial increase in the deep layer neurons that are being generated at the expense of the SATB2 expressing proportion. So here I'm giving you the scheme that by removing the deep layer postmitotic neurons, now you're prolonging the production of deep layer neurogenesis, and then subsequently the upper layer neurogenesis are followed by this deep layer neurogenesis. So this combined with other molecular functional analysis, we now have a good model for the temporal cell identity transition in the developing cerebral cortex, in which early on the progenitors express multiple transcription factors by default, and these cells are capable of adopting an early Cajal-Retz's fate. However, once the expression of FOXG1 kicks in, now the FOXG1 represses these multiple transcription factors, enabling the transition from these early-born Cajal-Retz's cells to deep layer projection neurons. And finally, once these deep layer neurons are postmitotic neurons are being produced, these themselves provide negative feedback, thereby enabling the transition between these deep layer to upper layer neuron transition. Okay, and then this is necessary for specifying the respective laminar subtypes from the Cajal-Retz's cells to the deep layer projection neurons to the upper layer projection neuron transition. So, and ours and many groups, including Sue McConnell's and Jeff Makulas, Nina Sestons, Yukiko Goto's. So we are now beginning to understand how the laminar subtypes are being instructed through a series of temporal codes that are instructed in the progenitors as well as the postmitotic neurons. However, as I mentioned in the beginning, this layer subtypes or the layer organization is not even throughout this cortical spatial organization. So what are the actual codes that are instructing the different thickness between the different areas? And as I've mentioned, these laminar differences in the neocortical cytoarchitecture are best represented by the expression of R beta, or the presence of these sensory input layer 4 cells. So the question comes down to how these common progenitors, which are residing in the neocortex, then adopt a different laminar fate, specifically among these motor versus the somatosensory area, in which the somatosensory cortex has the abundant layer 4, and the motor cortex seems to lack these layer 4 neurons. And you could think about multiple hypotheses. For example, there may be like increased cell death in the motor cortex during the production of the layer 4 neurons. Or there could be excessive proliferation going on in the somatosensory area during the layer 4 production period. Alternatively, there could be a fate switch in the cells initially adopt similar fates. However, there might be some transition depending on some kind of extrinsic or sensory inputs. Or finally, there could be some kind of a migration difference that differentially specify this area of cytoarchitecture differences. So we decided to tackle each of these problems by doing respective assays. And first of all, here I'm showing the whether cell death could contribute to the differences in the future motor and somatosensory areas. So if you look into the tunnel and annexin-5 staining at postnatal day 1 in the mouse cortex, you don't see much of the cell death going on during this cortical postnatal development period. And this is consistent with other studies that at least these cell deaths are not substantially contributing to some of the features, maybe at later, for example, in the critical period phase. And then secondly, if you look at the proliferation between these future motor and somatosensory areas using different proliferative markers like Ki67, PH3, or BRDU, none of these were significantly different among these embryonic future motor versus somatosensory areas. We also looked into the cell cycle exit rate, doing a pulse of EDU, and then sequential BRDU, and then collecting the cortex 30 minutes later. And the cell cycle exit rate, again, was not different among these two different areas. So Together, this shows that the cell death and proliferation do not contribute to, are not the main contributors to these areal differences. So then we decided to look into the two remaining possibilities about whether there's a fate switch or some migration differences. And you would actually see that assessing both of these hypotheses requires to follow the fate of the newly born neurons across these different spatial regions, and then see how these different neuronal precursors end up over the time. So for this, we again turn into this genetic labeling of temporal cohorts of layer neuron precursors and use this expression of neurogenin 2. If you look at the transcript of neurogenin 2, it's highly expressed in the glutamatergic precursor progenitor population. However, Takaki Miyata back then showed that the protein level of neurogenin 2 is highly upregulated during the cell cycle exit timing. So these neurogenin 2 high cells are no longer Ki67 positive. So we thought that this was a nice way to mark the newly born cells, the transient newly born cells, to do a sort of a genetic birth dating analysis. So Chihiro Nishiyama back then made an heroic effort to do a series of tamoxifen administration by crossing these neurogenin 2 CRE-R with the Rosette 26 stop TD tomato lines, and did tamoxifen and see how those labeled cells end up in the laminar positioning in the developing neocortex. And you could now see that by giving these sequential tamoxifen time series, you are now able to nicely recapitulate this inside-out generation of these positioning of these TD tomato positive cells. And you also would appreciate that this not only labels the layer 5 versus layer 4, but even within the sub-territories of these layer 4 neurons, somatosensory regions in the cortex. So now you can focus onto this E15.25 tamoxifen administration stage, in which you could mark the layer 4 neurons in the peak timing within these sublayers. So Naoto Ote and Chen Zhao has done some morphological analysis. And if you give tamoxifen at E15.25 and do the analysis of the mouse cortex at P21, you could nicely label this TD tomato positive cells in the aura beta layer 4 neurons. And this has a very nice spiny stellate morphology in these coronal sections, as well as if you look into the tangential section, you see these nice somatosensory barrels, which are followed by this spiny stellate having a very nice polar morphology in this region. So we use this E15.25 as a time point to label the cohorts of layer 4 precursors in the somatosensory cortex. And we first did an in vitro studies to see how this different future motor and somatosensory cortical-derived neurons behave. And what we found was that when we do the tamoxifen at E15.25 and then isolate the cells at E16.5 and then do a 10 days culture in vitro, which is equivalent to postnatal day 7 in mouse cortex, you see that even though these precursors have not received much contact for the thalamic cortical axons, they already have a feature that is represented by both the morphological pyramidal shell type in the motor cortex versus the spiny cell type in the primary somatosensory derived precursors. And these cells express respectively CTIP2 and also aura beta in the somatosensory cortex. So it appears that these regional identities are actually fixed pretty much early on during development, earlier than what people has appreciated. So there appears to be not a later fate switch, but a predetermination of these early precursors. So we finally looked into the possibility of whether migration differences could contribute. And for this, again, we used the same E15.25 tamoxifen, and they did the analysis. We labeled the cells and we sacked the cell cortex at E16.5. So you now see that the newborn TD tomato positive cells, which I'm sure showing here in green, is being generated from the respective motor versus somatosensory. And if you zoom in and do a time-lapse imaging, now you see that there is a rapid migration of these medial cortical neurons. I'll show you again in this medial proportion, whereas this lateral region, there is a pausing phase of these newly born neurons. So there's apparently a difference in the migration patterns of these medial versus lateral neurons. And because this is a time-lapse movie, just to show that this might be some kind of an artifact, we also did this analysis in vivo and sacked the cortex at E17.5. So you now see a substantial differences in the positioning of the newly born neurons in the medial versus the lateral, future motor versus somatosensory. And if you look at these positioning of these neurons at P7, now those neurons in the motor areas have occupied a more deeper positioning, whereas in the primary somatosensory, they are nicely aligned in the mid of the layer 4. So we also did a single cell analysis to look into the respective molecular candidates and data clustering, and found that there is a substantial differences in molecular repertoire in these posing cells versus these early rapid migrating cells in the primary somatosensory area, primary motor area. And we did a series of studies and found that there are some representative marker genes that are specifically expressed among these pausing cell populations. So finally, we have done a series of functional studies of each of these genes and found that, for example, if you introduce these genes that are represented in the pausing cell population and put that, force express that into the developing motor cortex, now you are able to recapitulate this pausing behavior of these migrating cells. So this is the cortex GFP expressing introduced cells, whereas if you introduce two of the genes ADGRL3 and TFAP2D, now you're seeing a substantial migration delay or a pausing behavior beneath this cortical plate. So finally, we now have some idea of understanding how the neurons that are generated from the common progenitors across these spatial areas adopt different positioning or cytoarchitectonic differences. And we think that these are partially due to the molecular specificity, which gives some instruction for these spatial migratory cues. And we think that this is one of the mechanisms that would explain how the cytoarchitecture differences arise, which was originally proposed by Brodmann back in 1909. And sorry, I'm kind of running out of time, but this study was spearheaded by Chihiro Nishiyama and Pei-Chan Ho, who now has her own laboratory in Taiwan. And this is now followed by Naoto Ote, and many of the people in the lab are looking into the cytoarchitecture difference in different areas. So thank you very much, and I'll be happy to take questions.
Okay, great. Awesome, Carina. It's an amazing talk. So
thank you.
Open for questions. So I had the first one. So amazing. The second part of the talk is the boundary separation. That's such a elegant mechanism. But so would you suggest that there be kind of autonomous mechanism for not so migrating fast or pausing at home? Yes. I was originally thought it might have some inhibition outside in the area where the neuron won't belong. Then, so what do you think? So
yes.
Why exactly?
Yeah, thank you. So yes, I think the original idea of these area differences in the cytoarchitecture is due to some, for example, the contact with the thalamic TCA axons. So that would probably provide some kind of a molecular cue. But even though if you consider that there is a cue that comes in from the extrinsic, cells like the thalamic neurons, you need some kind of an intrinsic cue to be able to attract those axons, right? And then now, when we do these single cell studies really early after the cells have just exited the cell cycle, we already see that there are molecular expression differences between these two areal populations. So, of course, we don't know exactly what are the upstream factors that, for example, the morphogen or secreted molecules that lead to these molecular differences early on in the development. But we definitely know that these genes are expressed quite early, which is able to, for example, give some kind of responsive cues to the surrounding environment, for example, the subplate or some kind of incoming thalamic axon. So that's actually exactly what we are trying to get in now. Yeah.
Amazing. You're suggesting the predetermined.
Right. Exactly. Right.
Yeah,
yes.
Really. Thanks. Yeah. Right. And more questions. Let's see. Should I do a kind of question? I saw the comments. And they.
Sorry, it's long. Yeah, sure, Corona, that's a really nice talk.
Thank you.
It's really beautiful study. So most of those studies in those cortical self-determination focus on those transcription factors. Do you have any insights about whether there are some extracellular signaling that could be involved in the self-determination in different areas of the cortex?
Right. So initially, when we were focusing on these intrinsic areal differences, we of course wanted to know what is the master transcription factors that may differentially, confer these identity. And we published a few years ago that at least the transcription factor COOP-TF1 and the antagonistic effect with FOXG1 are responsible for making layer 4 versus non-layer 4 cells. And we also tried to identify the upstream factors of these transcription factors. And we know that FOXG1 suppresses COOP-TF1. However, upstream of FOXG1, there are several EGR genes that seems to repress the expression of FOXG1 in the future layer 4 neurons. So there are definitely some, for example, maybe TGF-beta related or maybe like activity-dependent mechanisms in the that are instructing these transcriptional differences. However, we still need to go into a little more, fine scale functional analysis to be able to distinguish, or what's of course the very, very beginning of this areal identity the determination. Yeah, but that's a very, very important question. Yes.
Thank you.
Thank you.
Thanks, Josai. Any more questions? So I have a last quick question for the FOXG1 story. So you might already know. So FOXG1 is one of the critical gene for contributing to the Rett syndrome. So which also shares which part of MCB2 is also one of the major gene I studied for years. So what's your insight about is a reason FOXG1 mutation cause Rett syndrome similar with MCB2 or has some other mechanisms?
Yes. So FOXG1 at least is, of course, it's a master regulator for brain development, and then it's also a key gene that is perturbed in both, like for example, autism or schizophrenia. So we think if we consider FOXG1 being not just the master gene for specification, but also if it's switching these sensory input subtypes or the output, like the layer 2, 3, or layer 5 projection neurons, then we could view this in a sense that if the brain is sort of trying to balancing the input versus the output, FOXG1 may be in the positioning of trying to not get too much input, but also trying to balance the output. So we think that maybe these phenotypes seen in these syndromes could be of course, it's a very, very difficult question, but we can try to, dissect out what are the actual pathway contributing these behavioral differences in these patients. Yeah.
Yeah, great. A few more comments is because in the FOXG1 Rett syndrome mutations, most likely there are point mutations.
Right,
so
right.
I agree your hypothesis. There are point mutation will affect some aspect of this function, might alter the sensory or motor pathways, might lead to the disease phenotypes. Thanks.
Yeah, thanks.
All right. So thanks, Carina. Now, Aaron, to introduce the second talk.
Great. Thanks, Zilong. Great talk, Carina. It's a great privilege to introduce our next speaker, my colleague at Stanford, Dr. Miriam Goodman. Professor Goodman is a professor of molecular and cellular physiology. She's also the chair of the Department of Molecular and Cellular Physiology at Stanford. Miriam received her undergraduate degree in biochemistry from Brown University. She then did her PhD at the University of Chicago, where she used whole cell voltage recording of potassium currents in cochlear hair cells from turtles. She then did a postdoctoral fellowship at the University of Oregon, and here she started working on C. elegans. And at the time, there was a wiring diagram for C. elegans, but it wasn't really clear the function of neurons. And Miriam pioneered patch clamp recording techniques, getting them to work in C. elegans. And this has really opened up many new areas for the field. And following that postdoctoral fellowship, she moved to Columbia University for another postdoctoral fellowship with Martin Chalfie. And here she started studying mechanotransduction, again in C. elegans. She had a beautiful paper in Nature where she discovers a mechanism underlying gentle touch sensing in C. elegans by channel proteins. She took those proteins and reconstituted the channel activity in xenopusoocytes. She defined the biochemical functions of these proteins, MEC4, MEC10, and then another membrane protein called MEC2. And she showed that MEC2 functions to regulate MEC4, MEC10 ion channels. She then launched her own lab at Stanford, where she continues to use C. elegans and, importantly, hardcore biophysics and electrophysiology to define mechanisms of mechanosensation. She's won many awards, all of the neuroscience awards, I think. She was an Alfred B. Sloan Fellow. She was a McKnight Scholar Award winner. She was the Klingenstein Fellow. She got the Eppendorf and Science Prize in Neurobiology. She won the Landis Award for Outstanding Mentoring from the NINDS. So it's great, great privilege to welcome my colleague, Dr. Miriam Goodman, to present her latest work.
Oof. All right. Well, that's quite a thing to live up to. So I just want to start by saying that I'm really honored to have slipped onto the roster of NeuroZoom before it ends. And I'm grateful to Aaron and Zilong for creating and sustaining this event for nearly 3 years. I think it really has expanded our community and a great testament to the range of neuroscience that's been presented here. And we'll see a bit of that today. So we've heard a bit about how brains are built, and I'm going to try to tell you a bit about how our sense of touch works. So let's get started. I'm going to tell you how touch is like real estate. Well, how is it like real estate? Well, it's everywhere. Single-celled organisms to arthropods to mammals to plants have all evolved the ability to sense external cues or have a sense of touch. It has varied forms that serve a similar purpose. Mechanosensitive ion channels function as the front door to touch, and they come in a variety of shapes and sizes, as I'm illustrating here. They're found across phylogeny, and in many cases belong to superfamilies of proteins that include both mechanosensitive and non-mechanosensitive members. So we don't have like a molecular structure that we can look at and say either from amino acid sequence or a structural motif or an organization of how those subunits are assembled into a channel that says, hey, I'm a mechanosensitive channel. Instead, we need to rely on other approaches to try to understand which ones function as mechanosensitive channels and how they function and how mechanical force is delivered to those channels. Aaron mentioned the MEC4 channel, and I'm going to be focusing today on MEC4, and it's a member of this family over here on the right, which is the only family that is present and exclusively present in animal genomes. All of the others are present across many different kinds of organisms. So as we move forward, I'm going to try to convince you that location is as important for touch function as it is in real estate. So this movie here on the left shows you how a worm responds to touch. When that green light comes on, green circle comes on, this animal is receiving a tightly calibrated stimulus using a device that we built of 1 micronewtons to its head, and you can see that it's moving backwards. This avoidance behavior has kind of formed the foundation of genetics that allows us to identify genes that are required for this function, and that is how the MEC4 channel, the MEC10 channel, and the MEC2 protein that Aaron mentioned earlier were first identified, and later we used electrophysiology to link them to the activation of the neurons that express them. So we did that about 15 years ago, and I don't have time to show you those data, but I want to tell you more about how we think MEC4 works and how we think that mechanical force is delivered to these channels in their native environment in the animal. So on the left is a schematic of a worm showing the 6 touch receptor neurons that together they cover the entire worm's body from head to toe. If we take a look at the touch receptor neurons labeled with an antibody specific for MEC4, we see the MEC4 protein over on the right, localized to discrete puncta. And we have an idea, although not a direct structure of what MEC4 might look like from a structure of a relative from a vertebrate, and that's what I'm showing down below. The neurons themselves are embedded in skin cells, and that's shown here in this EM micrograph on the right. So at the center of this micrograph is a cross-section of the neuron, and those tiny little circles in the center are microtubules. If you're used to looking at synapses, you might think they're synaptic vesicles, but they're not. We know that's true because we have serial sections. And then the skin cell is around the neuron, and it's separated by an extracellular matrix. In that extracellular matrix, I don't know if you can see it, there are some dark electron-dense structures in there. And one of the things I've been very interested in trying to sort out in recent years is how are those things made and what are they made of? And it's notoriously difficult to link a structure you observe in an electron micrograph to the proteins that make it up. And I think we're making some progress on that goal that I'm going to tell you about today. So this is ongoing work that we started more or less at the beginning of 2020 and have continued through that time to try to link these structures. So one of the things we were curious to know about that we've looked at most recently actually is whether or not these, so in serial section reconstructions, these structures which are in the extracellular matrix, they're small planks, they're about a micron long. But what we didn't know from short serial section segments was whether or not those structures were continuously present all the way along the neuron, or if they were in discrete regions like we see for the channel. So we reached out to Mei Zhen and her postdoc Ben Mulcahy to take a look at the data sets they had generated in order to add additional connectomes to our knowledge and ask them to look at the touch receptor neurons in their connectomes. And what we found from looking at those very long, basically full animal reconstructions, is that in fact, these structures highlighted here in the red arrows are discontinuous, and their spacing is quite similar to what we see when we look at the channel itself. So having done that, we wanted to know more about the proteins that might make up those structures. And to make progress, what we did was we gathered strains that labeled conserved basal lamina or basement membrane proteins, tagged at their endogenous loci with amnion green, and then screened them to see if we could identify if any of these proteins were present in the touch receptor neuron. We're grateful to David Sherwood at Duke University for sharing many of those strains with us. And what we found from that screen were 9 proteins that were located along the touch receptor neurons and 3, LAM1, LAM2, and nitigen, that were distributed into puncta that had very similar appearance to what we see for MEC4. So we use these tools to quantify the distribution, and this is what this very This graph is showing you here. So from left to right, we have LAM1, LAM2, NIDIGEN or NID1, and MEC4. And what's showing down below is the average is the distance in between adjacent puncta for each of those proteins. And I think what you can appreciate is that those distances are essentially identical. So the statistics of the distribution of these proteins, 1 channel and 3 conserved basement membrane proteins, is the same along the touch receptor neurons. So what else can we learn with this tool? Well, we can do some live imaging and ask what happens to those puncta over time. And we were really surprised to see that pretty much they don't move at all. And if we bleach a punctum, so you can take a look at each one for MEC4, LAM2, and NIDIGEN. If you bleach a single punctum, you observe it for 5 minutes, it does not recover. The adjacent puncta don't dim. So this tells us that these puncta are pretty much held in place and they're not mobile in the membrane of the neuron in the case of MEC4 or in the extracellular matrix in the case of LAM2 and NID1. So then this led us to think, well, maybe they're actually part of a single complex. So we built lines where we tagged the channel and one of the extracellular matrix proteins in different colors. So here we're using scarlet and neon green to label MEC4 and NID1 on the top and MEC4 and LAM1 on the bottom. It's a little hard to see the ECM puncta because there's labeling in many, many other tissues that makes it harder for us to see that. But we can use these images to quantify the correlation, the co-localization. One thing I should have mentioned earlier is that a single touch receptor neurite is only 200 nanometers in diameter. So these are really tiny structures. So we need to be super careful drawing conclusions about whether or not we see co-localization at the light level, which is why we're using a statistical analysis over on the right. So there are 2 clouds of points there, one paired. So there we're looking at the intensity correlation in a single animal of our 2 channels, and another shuffled, where we shuffle the 2 channels between different animals in our data set where we don't see very high correlation. So this gives us confidence, in fact, that they are colocalized despite the fact that we're looking at a tiny structure. Okay, so far I've shown you that MEC4 is coalesced into puncta, and so are conserved extracellular matrix proteins, laminins, and nitigens, and that they're co-localized. How do they get this way? And when we think about basal lamina or basement membranes, we normally think about them forming continuous sheets that separate one tissue from another. We don't think of them as forming discrete structures like those plank structures that I showed you earlier in the talk. So to learn more about how they come to be, we turn to 3 proteins that were identified in 4 genetic screens, actually done by Marty Chalfie and his students in the early 1980s. These proteins are known to encode extracellular matrix proteins, and they're all known to be needed for touch sensation and also for the formation of the MEC4 puncta. And we asked what happens to the puncta and the ECM. In other words, what happens to laminin and nitigen in these mutant backgrounds. I'm only going to show you data for MEC1, but I will say that when we look at null mutants of MEC1, MEC9, or MEC5, we see a disruption both of the MEC4 puncta and of laminin and nitigen. So when these ECM proteins are missing, the basement membrane doesn't form these discrete structures anymore at the interface between the neurons and the skin cell. Furthermore, I want to tell you one more thing, which is MEC1 and MEC9 are made by the neuron, not by the skin cell. So this suggests that the neuron is making proteins, secreting them into the extracellular matrix, and those proteins may be actively reorganizing the basal lamina into structures that are needed for function. So I told you I was going to tell you about MEC1. And in fact, I'm going to tell you a lot about MEC1, and some of which is summarized here on this slide. So MEC1 is a very large protein. It has 15 domains in it, which are Kunitz domains. These are a conserved structural fold. They have the same fold as cis loop toxins that bind to many ion channels that are made by many different kinds of organisms, often as tools to paralyze prey. MEC1 is required, as I've mentioned, for normal MEC4 distribution, and it's also required for NID1 distribution. So that tells us that it's needed, but it doesn't really tell us kind of what it's doing. And to learn more, we took advantage of the similarity in the structures of these Kunitz domains and toxins to try to engineer some mutations at the terminal Kunitz domain. So this is the one all the way at the C-terminus end of it, and ask what happens to the organization of NID1 and MEC4 when MEC1 is present, but it has point mutations in this structure at the end. So what we found is, so this first allele up here is one that came out of the initial 4 genetic screens, and it codes a mutation in that 15th Kunitz domain. And then the others are mutations that we made using CRISPR. So might be easiest to look at the panels in the center, where I'm showing the distribution of interpunctum intervals in wild type in gray, and in the mutants in magenta for the distribution of NID1. So I think you can see that in those MEC1 mutants, NID1 becomes sparser. There are maybe fewer puncta. If you look over here on the right, this intensity quotient, correlation quotient I introduced to you earlier, is dramatically reduced in each of these point mutations. So MEC1 is present, MEC4 is present, NID1 is present, but they are no longer co-localized. Moreover, all of these animals have severe touch defects. So we are thinking about, sorry, let me skip. Oh, I am missing a slide. Look at that. That's fun. Let me see if I can find it. Give me 1 second. Okay. So one way that we're thinking about this is in this diagram over here on the right, where we imagine that MEC4 sits in the plasma membrane and it's connected to these defined structures made by laminin and nitogen in the extracellular matrix by means of this MEC1 protein, wherein this 15th Kunitz domain is actually connected to MEC4. We don't know that for a fact. That's our current model. So now let me back up a bit through a few slides again. Okay.
So, Miriam, the sharing stopped just now.
Oh my gosh, I'm so sorry.
That's
Thank
good.
you for letting me know. That doesn't help anyone. All right. So let me show you the diagram I just told you about, and then I'm going to show you some more data. Thank you for letting me know, Aaron. All right. Can you all see that?
That looks good.
Okay, terrific. So just to recap, MEC4 sits in the plasma membrane. We have these structures in the extracellular matrix. We believe that they're linked by MEC1 with that 15th Kunitz domain of MEC4 forming an interface with MEC4, of MEC1 forming an interface with MEC4. So let me back up a little bit and show you some more data. Okay. So at this point, you might be thinking, oh, that's kind of cool, but is this nano interface that I'm mapping out for you something that's specific to the C. elegans touch receptor neurons? And I'm here to tell you that it isn't. We see similar close opposition between sensory neurons in flies, in zebrafish, and even in humans, where in the epidermal layer, there are sensory neurons that actually traverse through tunnels in keratinocytes in the skin. So this interaction between somatosensory neurons and the skin cells that they innervate is something that is present across multiple animals and not specific to C. elegans or to the touch receptor neurons. So I told you I was, my mission here was to convince you that location was as important for touch sensation as it is in real estate, and also to circle back and connect to function. So one of the things I love about this particular sculpture is that the sculptor, this is a marble sculpture, understood that when a touch occurs, it doesn't occur only at the place of contact. So you can see the stretching and the deformation of the skin at the contact with each of the fingers in this particular sculpture. So we can also see that when we look at what happens when we are indenting with a biophysical tool, worm. And this is the same kind of tool that we use for doing electrophysiology, what happens to the worm's body. So you can see that it's indenting more where the tool is going in and deforming the skin a little bit at a distance. So when we push, and we push further and further, we get larger and larger currents, and that's shown both on the left and on the right of this particular slide. And we've noticed that we really didn't ever see this response saturate. And then as we started to think about that and to build up a biophysical model of how that might work, we've built a model that connects the force applied to the skin to the activation of individual channels as a function of the deformation of the skin at the point where we're pushing and at a distance. And using that model, we can ask, well, what is happening as we push further? Are we opening more channels, or is the likelihood that each individual channel gets opened increasing? So the panel in the center of this graph shows that pretty much from the moment we start pushing, our channel conductance, that is the probability of opening a channel, goes to its max level right away. However, the number of channels that's opened increases as we push. So what's happening is we're recruiting channels at a distance from this local stimulation. Now, circling back to what I told you about the extracellular matrix, what happens if NID1 is missing? I can't tell you what happens if any of the laminin genes are missing because all those animals, unfortunately, are dead. But the NID1 nulls are not. So what we find in the NID1 nulls is that MEC4 and laminin retain their organization as discrete puncta. They stay co-localized, but they're sparser. Okay? And when we look at behavior, we see a minor decrease in behavioral sensitivity in the NID1 nulls. We see a decrease in the amount of current that we're able to recruit in electrophysiology. And together, this tells us that when the puncta are sparser, we can still activate them, right? And the activation still increases with as we deliver the stimuli. But we never get quite as much current as we did at the beginning. So now I'm turning back to this diagram that I showed you earlier. And what we think is happening when we're pushing on the skin, we're deforming the skin at that location and at a distance. And as we do that, we're pushing these structures in the extracellular matrix off to one side. That's tugging on the MEC4 channel and opening it. And this is our current model, biophysical model of how we think this happens. And this is something that relies on the interface between the neuron and the skin cell that would be extremely difficult to recapitulate outside the animal. So where touch happens matters for how touch happens. And I'm at my end here, and I just want to make sure that I mention many of the folks who worked on this project. Their names are all highlighted in yellow. This team was led by Elke Das in the upper right-hand side, mention also Linjing Wang, who did all the electrophysiology, and Juan Cueva, actually, who has not been in my lab for a decade, has contributed EMs that are part of the story that I've just told you today.
Thanks so much, Miriam.
Okay.
And we're open for questions. Jin?
Hi, Miriam. That's wonderful talk. And I guess what I was sitting there thinking about several questions. One is a MAC4, I mean, how many subunits is there to form this MAC4 channel?
Mm-hmm.
And how many of this MAC1, the 15th repeats that needed
Yep.
to be there?
So the complete channel has 3 subunits. MEC4 is co-expressed with MEC10. So each channel has at least one MEC4 and one MEC10 molecule. We are actually unsure if each molecule has 2 MEC4 or only one MEC4 protein. And the current evidence suggests that there are 2 MEC4 proteins per channel and 1 MEC10. So that answers the question on the channel side as much as I can today. On the MEC1 side, we don't really know for sure how that might work, but from a biophysical standpoint, it's a bit easier to think about the mechanics if MEC1 is really just holding on to one side. Okay, so getting back to the stoichiometry of the channel, let's say hypothetically, instead of being attached to MEC4, it's attached to MEC10, of which we think there's 1. Do I know this is true? No.
Okay.
Am I excited to try to figure it out? Yes.
Cool. Thank you.
Other questions? I have one, Miriam. So I'm just wondering what decides how many MEC4 channels there'll be in the regular spacing. Like if you upregulate MEC4 levels, do they produce more, or do they all can sort of condense in the same regular pattern?
Yeah, so that's a really interesting question. So to kind of ask the experimental question that you've asked, so we've been able to look at the distribution of MEC4 proteins with many different markers. So the first image I showed you was of an antibody to the native protein. And the most recent data that I've showed you is M-neon green tagged MEC4. The M-neon green tag MEC4 is actually expressed as a single copy transgene and not from the MEC4 promoter. It's from a promoter that's a little bit stronger, so it's probably overexpressed. We also have another marker that's overexpressed. So when we look across these different ways of seeing the channel, we see the same statistics of the organization. So that seems to suggest that there's some mechanism regulating how many channels there are and where they are, okay? That's not related to the amount of MEC4 protein that's present. So then if we think about what happens in the NID1 null mutants, where we still have the puncta present, but they're reduced in number, we can begin to say, well, maybe that's a suspect for what determines how many channels are actually there. So we can imagine the channels are maybe being, one way that I think about it is that like there are parking places in the extracellular matrix that the channels then go and occupy. And it's not the number of channels that determines how many parking places there are.
Got it. Okay. Other questions? I've been getting lots of texts in the about what the sculpture is, so I'm just pasting into the chat the
Thank
link
you.
for the sculpture, so everyone can take a look at that.
Yeah.
Great talk, and good seeing everyone. Looking forward to seeing everyone next week.
Bye. Thanks, Miriam. Thanks, Carina. Great talks.
Thanks,
Yeah,
Zilong.
yeah.
Thanks, Carina.
Aaron.
Good to see you, Miriam. Bye,
Good to see you.
Zilong. See
Hey,
you soon.
Carina, can I ask you a quick question?
Oh, sure, of course.
So I noticed that the ADGRL3 is an adhesion
Yeah,
GPCR.
Right.
Have you thought about whether or not some of the migration speed might be a mechanical effect?
Yeah, so we have tried to look into if there is, for example, like the differences in the stiffness among the areas, but so far we haven't had gotten some significant results on that. And then on the other hand, when we look into the interacting molecules for ADGRL3, some of them are actually expressed in the, for example, in the cortical plate. So there should, of course, be molecular interactions. Whether that's, could be also interpreted in the mechanical, regulation, that's a very interesting question. We still want to kind of explore into it. Yeah.
Awesome. Yeah. So it could be, even if there's no stiffness difference, that there's a difference in the way the cells are adhering to the area. That could slow them down or speed them up.
Right. Yeah, that's an important point. Yes. No, thanks for the input. Yes.
Cool. Thank you.
Thanks. Great.
Bye, everyone.
Bye.
Thanks. Bye.